So now I'm just going to talk to you while you eat dinner about some basic principles of clinical trials to introduce in a conceptual way why we do what we do, and I hope this is okay. I think it'll be review for some of you, and for those of you who are new, maybe something new is in there. So why do we do clinical trials as opposed to other kinds of study designs? And the answer is this study design provides the most definitive answers about the effects of an intervention on measured outcomes. It's an experimental design. The reason it provides the most definitive answers is it avoids bias due to non-random self-selection of treatment. One of the things that happens in observational studies is that the people who choose to take estrogen are healthier than the people who choose not to take estrogen. When you go to do an observational study, you find that they're healthier, and you don't know whether that's attributable to the fact that they took estrogen or the fact that they were already healthier before they started taking estrogen. And in fact, in the observational epidemiologic literature, they have shown that the women who started taking estrogen were healthier before they started in a variety of ways. One of the things about experimental designs is that you need to have a comparison group. It doesn't always have to be a placebo group, but you need to have two groups. Uncontrolled trials, trials where you just do the intervention in one group, have the potential to provide a very distorted view of therapy. And in the literature, the studies that have lacked a control group, and these are pretty much older studies now, are much more likely to lead to enthusiastic recommendation of the treatment as compared to properly controlled trials. So all the trials in this flash will have a control group. Adequate sample size is something that we'll be after in each of these trials, and that's because hypothesis testing requires enrollment of a pre-specified number of study participants to reduce the role of chance to a certain level and to improve the precision of the results. During recruitment, though, we realize, and I'm sure you realize, that there's a tension between getting eligible participants enrolled quickly and getting the right participants enrolled. And by right, I mean the participants who are really committed to finishing the trial and being adherent in it. So there is that tension. We don't get rid of it. We just have to deal with it. Random allocation, and this is where we use a process that employs chance to assign people to study groups. This ensures that any observed differences between treatment groups are due to chance. It doesn't ensure that all the treatment groups are going to be equal, because they might be different. But it does ensure that the differences that you see are due to chance. It eliminates bias from use of judgment or systematic decisions that might lead to differences between the treatment groups. And it allows for use of standard methods of statistical analysis, and it requires consent. So you'll be consenting these women to accept randomization within a trial, whatever they should be assigned to. And we won't even know what they're assigned to, but they have to be willing to accept randomization. Blinding, neither the patient nor the treatment team nor the evaluator know which treatment the participant is receiving. That's what blinded means. It reduces bias from expectation that the treatment will be and or the placebo won't be effective. And in studies of hot flashes, Dr. Freeman's been really clear with us that the expectation that your hot flashes are going to get better is a big aspect of future symptomatology. In Miss Flash, only the computer knows for sure unless we ask. And you'll see how anticlimactic it is to randomize a woman in the computer, okay? This is a funny slide about, some people say that you shouldn't use the term blinded, you should use the term masked. So these are blinded versus masked. One guy has on a mask that blinds him, and the other guy just has on a mask. Whatever you want, we'll be giving you your mask tomorrow. If you don't like the one that's completely blacked out, you can cut holes in. Blinding there are variations, and just for fun, I put in this slide from one of Garnett's slideshows. Single blinding is when the participant doesn't know of the randomization assignment. Double blinding is when neither the participant nor the study staff interacting with the participant know the treatment assignment. And triple blinding is double blinding when the reports from the trial are presented to the monitoring boards with codes instead of labels, so they don't even know active versus placebo. They might get told A versus B. But I've also heard triple blinding described as nobody knows what they're doing. So we want to avoid that at the DCC. And this is double blind versus single blind, and you can see that this fellow on the left has a double blindfold on. Okay. So what do we do to protect the blind? And this will come up in your training. Treatment assignment is not revealed at the time of randomization. The computer simply says randomized. Yippee, you randomized the person, they were eligible, you entered all the data that you had to do, and it'll say randomized. And that's it. That's all it's going to say. You go somewhere else, and it will tell you what number on the medication bottle to give that participant. And the computer will dictate always which bottles to dispense. They'll be in a box that's carefully architected by Fisher and either sent to the pharmacy that you tell us to send it to, or put into locked storage in your clinical site, whichever method you use. Evaluators avoid discussing guesses about treatment assignment with participants, and I imagine this is something that you just have to be a little bit vigilant about. You don't want to talk to people about what they think they're on if you can avoid it. You don't want to ask them questions that might reveal their treatment assignment to you. Although we'll be monitoring for symptoms and safety, and they will be answering questions on an individual level and entering them into the database. Everybody will be doing that. In some cases, the role of evaluation of outcomes is kept separate from clinical care. And this is what we do to protect the blind when, for example, taking the study medicine produces an observable sign. So like in an estrogen trial, a certain proportion of the women assigned to estrogen will start to bleed. And if the evaluator who's collecting data from that person knows that she's postmenopausal and started to have periods again, or started bleeding, she's going to know what treatment assignment that woman is assigned to. And in those cases, we separated clinical care from evaluation of outcomes. Data accuracy and objectiveness. Here the goal is to instruct women to complete forms and collect other data in the same way, standardization. So we'll have a procedures manual, and we'll be training you to give the same instructions for how to collect the data in the trial. And the steering committee has discussed various aspects of this in depth, I can tell you. When you give that hot flash diary, the instructions are going to be really important. And we've discussed so many details about this. You know, some women will want to, Dr. Jaffe said some women will want to put pennies on the bedside, and every time they wake up with a hot flash, move a penny over. And in the morning, they just count the pennies that they've moved and say, that's how many hot flashes I have. Other women will just wake up and say, oh, I think I had this many. And will want to try and standardize what the women are doing so that we're getting the same kind of data from everybody. It's important to keep missing data to a minimum. And so you'll see in the database that it will tell you when data are missing. And it will tell us when a certain amount of data are missing. And it will ask you to go get it if you can. Computerized data capture is designed to reduce errors of commission and omission. So it's designed to tell you if a field isn't valid. It'll say, that won't work here. You've got to come back with an in-range number. And it'll tell you what you're missing on that participant. So let's talk about adherence and retention in principle, and then tomorrow you'll hear less about it, I think, but you'll see that some of the procedures in the follow-up visits are designed to measure adherence and to promote retention. The definition of adherence is the degree to which the woman follows the recommended behavior in the protocol as prescribed. What percentage of their pills do they take? To what degree do they follow the prescribed diet or exercise or other requirements? And for us, adherence is also, they've got homework to do. They've got to fill out those hot flash diaries. And that's gonna be absolutely critical in all of these trials.